Hello and welcome back. Last time we talked about infection basics, how viruses get in and out of a host. For the next two lectures, we're going to talk about host defenses against infection. And if you remember anything, please remember one thing, and that is most of the symptoms you feel when you're infected with viruses are a consequence of host immune responses. Today we're going to talk about the first lines of defense against viral infections, what we call intrinsic and innate defenses. Here's my view of host defenses. I view host defenses as a series of barriers that viruses need to surmount or overcome in order to successfully infect a host. And success is defined as replicating and spreading to a new host. We start today with a consideration of the first three walls, and that's anatomical and chemical defenses, intrinsic defenses, and innate immunity. Anatomical and chemical defenses are always present. These are basically physical barriers like mucus, saliva, stomach acid, tears, skin, scabs, defensins. And these are all things that are present in us and on us and are aimed at preventing viruses from infecting. So, for example, mucus in our respiratory tract traps a lot of viruses, and we have a mucociliary elevator that brings the mucus from the tract upwards where we can swallow it. Another example would be skin. As we talked about last time, skin is dead. The outer layer of skin is dead. So when viruses fall on the skin, they can't infect. And the skin happens to be covered with defensins. These are proteins largely made by our skin microbiome, our skin bacteria. And many of those block virus infection. All right, so those are anatomical and chemical barriers. They're physical barriers that are always present. Then we move to intrinsic. So these barriers, these anatomical and chemical barriers eliminate many viruses, but some overcome them. And then they encounter the intrinsic defenses. And these are also present immediately. In other words, they can be activated very quickly. And they include autophagy, apoptosis, microRNAs, and CRISPRs. We'll talk about some of these today. If viruses overcome intrinsic defenses, they then encounter innate immunity, which takes anywhere from minutes to hours to be engaged. And this comprises natural killer cells, complement, antigen-presenting cells, neutrophils, and cytokines. And this, again, eliminates most viruses. But if some happen to overcome innate immunity, they will acquire acquired immunity, which takes hours to days to engage, and we'll talk about that next time. At every step here, viruses have evolved ways to overcome these defenses. So once again, today we're going to talk about intrinsic and innate defenses. Intrinsic defenses are always present. Defenses that are inside the cell, like apoptosis, autophagy, RNA silencing, and antiviral proteins. Then we're going to talk about the innate immune system, which is induced by infection. Next time we'll talk about the adaptive system, which is tailored to the pathogen and which requires more time to be engaged. Let's talk first about intrinsic defenses. A few examples. One is RNA interference. This is a form of intrinsic defense that is certainly present and has been demonstrated in plant and invertebrate cells. And it consists of a system wherein an incoming virus either has double-stranded RNA in its genome or makes double-stranded RNA during replication. That double-stranded RNA is recognized by a nuclease called dicer, which chops it up into short stretches of RNA. One of these strands of RNA, called small interfering RNA, then forms a complex with proteins in this cell called RISC. And one of the enzymes in that complex is AGO or Argonaute. This whole complex, the RNA plus the proteins, will then engage new viral RNAs that are produced in the cells. The siRNA will provide specificity by base pairing with the viral RNA, and then the Argonaute protein will degrade that incoming viral RNA. In other words, this system uses incoming viral RNAs as a template to produce a silencing RNA that will degrade more viral RNAs. Whether or not this system is present in mammals is controversial. A number of laboratories have been working on this. Some feel it's present, some feel it is not present in mammalian cells, and one of the ideas is that we don't need it because we have other forms of immunity that have evolved. So this is an ongoing area of investigation. Now, viruses do encode countermeasures to most steps in this RNA interfering pathway. As you will see, at every step that we discuss, I'll have a countermeasure word on each slide to remind you. And I'm not going to go into the detail always of the countermeasures, but to remind you that viruses can antagonize every step of these pathways. If they couldn't, they would be eliminated. There would be no viruses. Another interesting intrinsic defense is an antiviral protein in the host cell called APOBEC3. APOBEC stands for apolipoprotein B mRNA editing catalytic polypeptide, and this is a protein that we recognize as being antiviral for HIV-1, shown here in orange. So this would normally be incorporated into the HIV genome. So let's look at the bottom here first and see how that would happen. Here's APOBEC3G being incorporated into the budding virus, and you can see the orange ovals are now in the virus particle. So these APOBECs are in the virus particle. When this virus particle then infects a new cell, APOBEC3G causes mutagenesis because it is a deaminase. And that's shown in this little gray inset here. So remember, when the virus infects cells, the viral RNA enters the cell, remains complexed in the cytoplasm with reverse transcriptase and other proteins, viral proteins. Reverse transcriptase makes a minus strand DNA, and that is then the substrate for deamination by APOBEC. And you can see the consequence of removing an amine from a C, you convert it to a U. Then when you make double-stranded DNA, that U is copied to form an A. So normally it would be a C, which would have been copied to form a G, but here it's an A. So we have G to A mutations, and so that happens throughout the viral DNA. It's extensively mutated, and it's rendered non-infectious because there are too many mutations to be compatible with viral infectivity. People have called this protein a weapon of mass deamination, WMD. What I've shown at the bottom here actually doesn't happen with HIV because the virus encodes a protein called VIF. And VIF targets APOBEC3G for degradation by the proteasome. So here is VIF in blue here, and what is shown here is it's part of a large protein complex that ends up interacting with APOBEC3G and directing it towards the proteasome. What happens is it is ubiquitinated. These ubiquitin residues are added to APOBEC. APOBEC with the ubiquitins are sent to the proteasome, and APOBEC is degraded. The little orange bits here are degraded APOBEC. So in cells infected with wild-type HIV, there's no APOBEC3G in the virus particle. You have normal reverse transcription. It's only if you make a VIF minus virus is APOBEC incorporated, and does it cause deamination. So VIF is an antagonist of APOBEC3G. Another form of intrinsic defense is epigenetic silencing. We've talked briefly about this before. Epigenetic means modification, not of the DNA, but of something else to affect gene expression. And as we know, as we've discussed before, DNA in our nucleus, whether it be cellular or viral DNA, is in the form of chromatin, where the DNA shown here in blue is wrapped around nucleosomes, which are composed of histone proteins. When this chromatin is very compact, it's silent. Genes can't be transcribed very effectively. And I've told you before that when chemical modifications of the histones or even the DNA occur, this can loosen up the chromatin and allow transcription. So here we're showing acetylation of histones that opens up the chromatin and allows genes to be transcribed. That is, it allows mRNA synthesis to occur. So the compaction of chromatin is a form of epigenetic silencing. It can be achieved by removal of acetylated histone tails. And in cells, when viral DNAs get into the nucleus, they are typically wrapped around chromatin and compacted to silence them. And this often happens in what we call PML bodies. Here's a immunofluorescent staining of a virus-infected cell where it has been stained with an antibody to a component of PML bodies. And you can see these green punctate dots in the nucleus are areas where these proteins are focused, and they lead to deacetylation and silencing of chromatin. So again, the idea is that a virus infects a cell, the DNA goes in the nucleus, it's silenced by these mechanisms, and one of them is incorporation into these PML bodies where, for example, acetylated histone tails are removed. Viruses encode countermeasures. For example, human cytomegalovirus encodes a protein called 71 that causes degradation of a cell protein called DAX, which is needed for deacetylation. In other words, deacetylation will silence the viral DNA, so the viral genome encodes a protein that interferes with deacetylation. And Epstein-Barr virus and adenovirus also encode proteins that affect PML protein localization. And so it will disrupt the formation of these PML bodies, so the virus DNA is not included in the PML bodies and can't be silenced. Another form of intrinsic defenses. There are many others. One more is called apoptosis, programmed cell death, of course. Cells respond to stress in a variety of ways. One is that they kill themselves, suicide, programmed cell death. Here's a normal cell. If it's stressed, there are many ways that stress come about. And one of them, of course, is virus infection. Virus infection triggers apoptosis. The cell responds by peeling off bits of its cytoplasm. These are called apoptotic bodies. And eventually the cell will die. Many other changes occur in this cell. But the host actually monitors apoptosis. These apoptotic bodies that are released from cells are taken up by macrophages and other antigen-presenting cells like dendritic cells. And they take a look at them and say, is there any foreign protein in here? And if this stress has occurred by some normal cell mechanism, the proteins here will be recognized as self and there won't be any response. But if a virus has induced apoptosis, the macrophages will then initiate an adaptive response, as we'll talk about later. The response of apoptosis is in order to kill the infected cell. The idea being, if we can detect virus infection early in a few cells, we can have them die and they won't be able to produce additional infectious viruses, and that will stop the infection. Here are some viral regulators of this process, apoptosis, because again, viruses encode antagonists of all these immune defenses. And you can see on the left is a cellular target, and on the right, in the middle, is the virus involved. Here's the viral gene and the function. I don't need you to know the details here, except to realize that at every step of apoptosis, these are all proteins that are involved in the apoptotic mechanism. Viruses encode antagonists. So, for example, BCL2 is a cellular protein that regulates apoptosis. Viruses encode a homolog of it to interfere with the initiation of apoptosis, and lots of other mechanisms here as well. And you can see, because so many different viruses are encoding inhibitors of apoptosis, how important it is to antagonize this for a virus to successfully replicate. We recognize that there are ancient forms of intrinsic defenses in archaea and bacteria, and that's, of course, CRISPR, the CRISPR system, which has become very famous because it's being used in many interesting ways for gene therapy and other purposes. And as a consequence, there's been a lot of litigation as to who owns the patents on CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. I have trouble saying it myself. And these are defenses, as I said, in archaea and bacteria. It's been found in a lot of archaea, half of bacteria. And in terms of viral defenses, Can also be a defense against plasmid DNA coming into cells. Here's a bacterial cell. It's being infected by a bacteriophage. The bacteriophage has injected its DNA into the bacterium, and this viral DNA would go on and replicate. But what's happening here is pieces of it are produced, small pieces are produced by the CRISPR system, and these are integrated into host DNA. And should this host survive, these DNAs incorporated in an array, and that's in part where the name CRISPR comes from, these are a memory of previous infections. So if this cell is infected again by the same virus, these repeated integrated pieces of the viral genome are transcribed into cRNAs, which then together with a protein will degrade the incoming viral DNA, an ancient intrinsic defense. Of course, this has been repurposed now to use these activities to edit genes and so forth, and very exciting research possibilities. Let's take a quiz. The question is, intrinsic defenses are always present. Which of the following are included? Antibodies, T cells, epigenetic silencing, skin, or mucus? I'm going to tell you the answer now, so if you want to pause and think about it, go ahead. The answer, of course, is epigenetic silencing. That's an intrinsic defense. Antibodies and T cells are adaptive, which we'll talk about next time. Skin and mucus are those physical and chemical barriers. Let's talk about the innate immune system. If viruses have overcome anatomical and chemical barriers and intrinsic defenses, next they encounter innate immunity. This is a system activated within minutes to hours after infection. It comprises cytokines, sentinel cells like dendritic cells, macrophages, natural killer or NK cells, complement system. This can eliminate many viral infections, but if the viruses are overcoming innate responses, the innate immune system will inform the adaptive immune system. Hey, I need help. I can't deal with this on my own. We'll talk a little bit about that talk between the two systems today and next time. How does this innate immune system recognize microbes and not itself? This, of course, is the key question. At one time, we suspected that there might be a system, but we didn't know exactly what it was. It all starts in 1980 when two investigators, Christian Nusslein-Volhard and Eric Wieschaus, they're working on Drosophila. And what they're doing, something which was completely novel at the time, they were asking what genes are involved in development of the fly, Drosophila. And they were making fly mutants and looking for phenotypes and seeing what genes were involved. And one day they found an interesting phenotype that made very unusual flies. And the story goes that Nusslein-Volhard was looking at the fly under the microscope, and she called Wieschaus over and she said, "Das war ja toll," which is like amazing in German. And so this gene that was mutated in that particular fly and gave the phenotype that got her very excited was called the toll gene. And then 15 years later, they got a Nobel Prize for that. Very important work. And 16 years later, the toll gene was found to have a role in immunity of flies. Remember, it's first identified as a gene involved in development of the fly. And then a year later, these genes were found in mammals, and they were called toll-like receptors. And this is really the birth of our understanding of the innate immune system, which has since gone on far beyond toll-like receptors. But this was the genesis. Now, the toll-like receptors, we have many identified since then. These are called TLRs, or pattern recognition receptors, PRRs. And they're, as the idea goes, they're responsible for identifying a pattern in a molecule, whether it be DNA, RNA, or protein, or lipoprotein, that's not self. It's a foreign substance. So here on the top left, here are the various toll-like receptors from 1 through here on this particular list is 10, but there are more. And you can see these are transmembrane proteins. There's the plasma membrane. There's an extracellular portion, which is made up of leucine-rich repeats. So the little tan box is a leucine-rich repeat. And you can see the toll-like receptors are made of different numbers of those. There on the right is the original toll protein from Drosophila. It was called Spätzle. And each of these leucine-rich repeats can be seen in this structure. This is the X-ray derived structure of toll-like receptor 3, TLR3 extracellular domain only, so not the transmembrane or cytosolic domain. You can see each of these strands is a leucine-rich repeat, so there are many of them in this particular TLR3. And the way it actually looks in the membrane is shown on this model here. You can see the curved extracellular domain, again, composed of leucine-rich repeats. There are two of them. This is acting as a dimer, binding an RNA. You can see the double-stranded RNA in there. Transmembrane domain here, and a cytosolic domain as well. The cytosolic domain is shown in the diagram at the upper left as well. That's very important. It is involved in signaling. It's called the TIR domain, or the toll IL-1 receptor domain. On the right in this table are summaries of the toll-like receptors that have been discovered and what they recognize, microbial macromolecular patterns. TLR7 and 8 single-stranded RNA, TLR9 CpG DNA, that is C and G together, or unmethylated CpG oligonucleotides, and so forth. So this is the idea that these recognize molecular patterns in microbes that signal the cell that something foreign is present to initiate a response. So let's see how this recognition happens. There are a couple of basic ways. Some toll-like receptors are on the cell surface, and they can recognize viral proteins. And of course, these also recognize bacteria, but we're going to focus our discussion on viruses, of course. Many of the toll-like receptors are in the endosome, as you can see here. So viruses, remember, bind receptors on the cell surface. They're taken up, many of them are taken up by endocytosis. And there, the nucleic acid may be sensed by a variety of toll-like receptors. And here you can see TLR3, 7, 8, and 9 in the endosome sensing various kinds of RNAs, double-stranded RNAs, single-stranded RNAs, which are not present in the host cell. And so this signals them as foreign. We don't typically have double-stranded RNA in our cells. We don't have single-stranded RNAs with 5' phosphates. Usually they're capped, they're messenger RNA, so that would be detected as foreign. We also have cytosolic sensors of viral nucleic acids. Here is one for double-stranded RNA. This can be sensed by a cytosolic protein called RIG-I. We talked about PKR, which is activated by double-stranded RNA, produced during replication of various viruses, leads to phosphorylation of VIF2 and shutdown of host cell translation. But this also feeds into these pathways that the other sensors feed into. And the one unifying theme of all this is that when foreign nucleic acids are sensed, whether it be in the cytosol or within endosomes, this sensing initiates a signal transduction pathway. So for example, we see here in the endosomal toll-like receptors, there's a cytosolic domain which is interacting with another cell protein, and the engagement of a ligand in the form of one of these RNAs initiates phosphorylation cascades. The end result is that transcription factors are phosphorylated, and then they go in the nucleus and turn on an expression of innate immune genes. And these include cytokines of various sorts, TNFs, interleukins, and interferons. And the main transcription proteins that are involved are IRF3, IRF7, and NF-kappa B. And these have to be modified in some way, 3 and 7 by phosphorylation, to get into the nucleus. And this transcription that results produces interferons and cytokines, which have antiviral properties, as we'll see. DNA can also be sensed. There are a number of different pathways for sensing DNA, and one of them is shown here, which is a major pathway. And here we see a DNA virus being taken into the cell. These DNA viruses typically end up putting their DNA into the nucleus. But the idea is that in some cases, DNA may leak out of the entry pathway. So you have some DNA in the cytoplasm, and that's not good. DNA shouldn't be in the cytoplasm. It's confined in the nucleus. So when DNA appears there, it is sensed by a protein called cGAS. And cGAS stands for cyclic GMP-AMP synthase. It's an enzyme which takes, when it senses DNA, it then takes a molecule of ATP and a molecule of GTP and makes a cyclic version of them. It's called cyclic GAMP. And you can see the yellow and the blue indicating that these ATPs and GTPs have incorporated, but they are incorporated as monophosphates and in a cyclic fashion. The structure of this molecule is shown here. On the left is the GMP residue, one phosphate left from the GMP. On the right is the AMP residue, one phosphate left. And you can see they're joined by these cyclic bonds. That compound, cGAMP, is then recognized by a protein in the ER membrane called STING. And STING binds cGAMP. STING with cGAMP then moves through the Golgi, and then it interacts with a series of proteins that end up initiating signaling pathways. Transcription factors move into the nucleus, and then you have transcription again of genes that are part of the innate immune response, which we'll talk about in a moment. And these proteins end up phosphorylating IRF3, for example, which then forms a dimer and gets into the nucleus. IRF3 then binds promoter regions in order to activate transcription. And here we have phosphorylation of a subunit of NF-kappa B, leading to release of NF-kappa B, entry into the nucleus. It also combined the promoter region. So that's how DNA is sensed. And again, whether you're sensing DNA or RNA, the end result is transcription of innate immune genes in the nucleus. Of course, viruses encode modulators of sensing. And here's one example of how Epstein-Barr virus, a herpes virus, produces proteins that interfere with sensing by toll-like receptors. So again, TLR2 can recognize viral proteins on the cell surface. When that happens, it initiates a signaling transduction pathway, which eventually will result in NF-kappa B going into the nucleus and the production of cytokines, which will then be involved in inhibiting viral proliferation. And shown also in this image is double-stranded RNA sensing in the endosome by TLR3, which leads to signaling also fitting into this pathway. Epstein-Barr virus encodes a number of proteins that antagonize this pathway. So here, for example, TRAF6 has to be ubiquitinated in order for it to proceed through this pathway. So does this protein NEMO in order to continue the signaling. And Epstein-Barr virus protein BPLF1 blocks, you can see that's shown by the red line, it blocks ubiquitination of these two proteins, so effectively inhibits signal transduction. Entry of NF-kappa B depends on degradation of I-kappa B subunit. That's blocked by BPLF1, effectively leaving I-kappa B to prevent NF-kappa B entry into the nucleus. So you can see multiple points of intervention of BPLF1 in this pathway, which will inhibit aspects of sensing so that antiviral cytokines are not produced. Our next question, which of the following allow the innate immune system to distinguish microbes from self? Cytoplasmic helicases and toll-like receptors, antibodies, apoptosis, APOBEC, or all of the above? If you'd like to pause now, I'll give you the answer to this question. The answer, of course, is A, cytoplasmic helicases and toll-like receptors. That is part of the innate immune system. Antibodies are not. Apoptosis and APOBEC are not. Antibodies are adaptive responses, of course. Apoptosis and APOBEC part of the intrinsic responses. It is the helicases and TLRs that allow distinguishing microbes from self. Let's talk about some of the products that are induced upon recognition of foreign substances by either toll-like receptors or any of the other sensors. We've talked just about a few. There are more that we haven't talked about, but the principle is the same. One of these products is interferon. This was discovered in 1957 by two investigators, Isaacs and Lindenman. What they found is that if they took chicken cells in culture and put non-infectious influenza virus on them, so in other words, the virus didn't replicate, it was inactivated either by heat or by UV irradiation. They put this virus on cells, the cells supernate, and after a period of time contained a substance which, if placed on other cells, would inhibit infection. And they discovered this reproducibly, and they called it interferon. Great name. because it interfered with the replication of viruses. And now we know that interferons are a whole class of substances made by virus-infected cells. They can also be made by uninfected cells in response to products released from cells. What does that mean? Now let's take an example of an infection at an epithelial barrier. Our virus is binding here, it gets into the cell, it's replicating, it's sensed. Infection is sensed by the sensors we've talked about. These cells will then produce interferons among other cytokines. So interferon is a kind of cytokine, and that's shown by the little red dots here. So these cells are producing interferons and other cytokines. They're attracting immature dendritic cells to these regions. And the dendritic cell is becoming activated and will then go to the lymph node, as we'll see later on. These dendritic cells can also pick up products released from these infected cells. If these infected cells apoptose, the blebs that are released from cells can have, say, viral RNA in it. That can be picked up by the immature dendritic cells. And then these dendritic cells will sense it as far, and they'll make interferon. So they're not actually infected, but they're still able to produce interferon because they sense the product of infection released from the infected cells. 3 classes of interferon we recognize important in virus-infected cells. Type 1, there are 13 different kinds of type 1 interferon, 1 type of type 2 or interferon gamma, and 3 types of type 3 or interferon lambda. These interferons are recognized by distinct receptors on the cell surface. So in this picture, we're showing you the receptors for these 3 different types of interferon. Those are transmembrane proteins, multimers, as you can see. And then the interferons themselves are shown by these little molecules that are bound. For type 1, it's this purple oblong molecule, and for type 2, it's these 2 purple spheres. And for type 3, it's the red oblong molecule. And these are binding specific receptors called IFNAR1 and 2 in the case of type 1 interferon. Those are the 2 subunits. And then for the type 2 receptor, again, 2 distinct subunits, and as well for the type 3, 2 distinct subunits. You can see here, these are transmembrane proteins. They have a cytosolic domain, which seems to be binding other proteins. And this means, of course, that the binding of interferons initiates a signal transduction cascade. These are protein kinases that are activated when interferons bind, and they will lead to the phosphorylation of other proteins and eventual downstream effects, as we'll see. When interferon is made, it's typically within hours of infection. So a virus infects the cell. The nucleic acid or the protein is sensed rapidly by the sensors we've talked about. The result is transcription of the interferon genes in the cell nucleus. And this declines. There are mechanisms in place to turn it off because interferon is toxic to us. So we only want to have bursts of interferon, enough to get rid of virus infection. The symptoms of producing interferon are global, as you'll see in a moment. The interferon that's produced is secreted from cells. You can see it's being secreted from a cell here in the upper right. And then the interferon binds to interferon receptors. Depending on the type of interferon, it will bind to a distinct receptor. Here we have type 1 interferon binding to the IFNAR1-2 heterodimer receptor. That binding leads to a signal transduction pathway involving phosphorylation of other proteins. Eventually, the entry into the nucleus of ISGF3, which binds to interferon response elements in genes and turns on the synthesis of over 1,000 mRNAs. These genes are called interferon-stimulated genes, or ISGs. We don't know how most of them work. We do know how a number function and how they're antiviral, and I'll show you that in a moment. This interferon response element is in part comprised of promoters, this is amazing, that have been acquired from retroviruses. Now remember, many retroviruses lead to the establishment of what we call endogenous retroviruses. The provirus is integrated into the germ cell and it's passed on to offspring. So humans have about 8% of our genome composed of retroelements, including endogenous retroviruses. And to refresh your memory, we have an ancient retrovirus that infect human ancestors many years ago. A germ cell established a provirus, LTRs with the viral genes in between. And over the years, these endogenous elements can jump around. And what has happened is that the LTRs have been repurposed to serve as promoters of innate immune genes, in particular interferon-stimulated genes. If we go back to this slide, when this transcription factor enters the nucleus after the cell has been engaged by binding interferon, it binds to gene promoters of interferon-stimulated genes, or ISGs. These promoters, many of them are comprised of the LTRs from retroviruses. So these are occupying between 6 to 14% of mammalian genomes. You can see all these mammals here. You can recognize humans, lemurs, rodents, bats, etc., all the way up to elephants. They all have these elements upstream of their interferon-stimulated genes that are derived from retroviruses. So just another example of how so much of our function has been repurposed from retroviruses. We talked about the placenta. Here's another example, which is the innate immune response. If you want to hear more about this, you can go listen to the people who discovered this on TWIV 382. Now, let's talk a bit about the function of some of these interferon-stimulated gene products. And again, there are over 1,000, and most of them we don't know how they work, but people are investigating them one by one. And this is a very cool one called Tetherin, or CD137. Tetherin is a protein that is, again, induced by interferon. So a cell's infected with a virus, the cell produces interferon, it binds to interferon receptors, induces a whole bunch of genes, and this is one of them. It's a protein produced on the cell surface. Interesting, it's attached to the membrane at both ends of the protein, and what this protein also does is it insinuates itself into viral membranes. So viruses produced by budding will contain copies of this Tetherin. And here is a diagram showing HIV budding from a host cell. And Tetherin forms dimers. So one copy of Tetherin in the host cell will interact with another copy of Tetherin in the growing bud of this virus in multiple ways, as you can see here. And it holds them to the surface. That's why it's called Tetherin. It tethers the virus particles to the surface. You can see a big chain of budding virus particles here in the diagram. And on the right is a cell showing this phenotype. The cell is at the bottom, and the viruses are stuck to the surface via these Tetherin chains. You can see a nice blow-up of that there. Now, of course, there's an antagonist encoded in the HIV genome. It's called VPU, which prevents Tetherin from getting to the cell surface. And so that's why HIV replicates, because it has this protein that has evolved to have this function of antagonizing this particular interferon-stimulated gene product. Very cool. Another interferon-stimulated gene product is called IFIT1. That stands for interferon-induced protein with tetra-tricopeptide repeats 1. This is a very interesting molecule that binds RNAs lacking 2' O methylation. What is that? Here on the upper left is a picture of a cap structure on a viral RNA. Remember, mRNAs and many viral RNAs are capped. The cap is a reversed 5' to 5' linkage of that first base to the first base of the RNA. And in addition to the cap, these structures are methylated here in yellow, you can see. And this is 2' O methylation, which simply means the methyl is attached to the 2' oxygen on the ribose ring. So the IFIT will interact with the cap, but of course all of our capped mRNAs would be recognized. So in addition to recognizing the cap, it will also need to see that there's no methyl here. And if there's no methyl present, it will bind. Apparently the methyl will interfere with binding. For example, what's the function? What's the consequence of that? Here's IFIT1 binding to the cap of a mRNA. And again, it's got to lack 2' O methylation for IFIT1 to bind. And that binding will block the cap binding protein from binding. And that stops protein synthesis. So the idea is if this is a viral RNA, you stop it from being translated. And you distinguish between viral and cellular because cellular mRNAs have these methyl groups and they won't bind IFIT1. It's pretty brilliant, pretty amazing evolution. The structure of the cap bound to IFIT1 has been solved, and that's shown at the right here. And there are two versions. The IFIT1 protein is shown in these cylindrical images, and this is bound to M7G, which is the cap portion here. So M7 is the methyl 7G, and that's shown in red. You can see there's a tunnel in which this binds, and a space-filling view is shown at the right here, and you can see the M7G bound in this tight tunnel. It won't fit in this tunnel. The M7G won't fit in the tunnel if the methyl groups are present. And so that's the distinguishing factor. It's a very small tunnel, and if the methyl groups are present, the cap won't fit in there. So that's how it distinguishes host from viral mRNAs, because it turns out many viral mRNAs don't have these methyl groups. But do you think that viruses can get around this? Of course they can. Here's some ways that they can. It's called escape from IFIT1. Influenza virus takes caps from host cell mRNAs. Remember, the primer for mRNA synthesis of influenza virus is a cap plus 10 or 13 bases from the host cell. Of course, these are methylated because they're derived from the host cell, so IFIT1 doesn't work. But coronaviruses don't even have caps. They don't care about IFIT1. All right, so those are pretty clear. Here's some other very clever ones. Some viruses encode a 2' methylase. The RNAs are methylated, the caps RNAs are methylated, so no binding to IFIT1. Some viruses steal host 2' O methylases. There they are. So again, IFIT1 doesn't work. And finally, some viruses have a stem loop structure right near the cap, which prevents IFIT1 from binding. Lots of ways to get around IFIT1. And again, if viruses didn't encode antagonists of all these defenses, they wouldn't be around. I mentioned this earlier, but let's mention it again. The interferon system is dangerous. In fact, this person here who's suffering from some kind of fever and chills, infection, maybe flu, well, most of his symptoms are caused by interferon. Remember, interferon induces the synthesis of many gene products. Most of our cells have interferon receptors, so in principle, you get infected, most cells in your body can respond to interferon. And that's why you get flu, you feel generally crappy. You have muscle aches, fever, tired. What is this, from a virus infecting my lungs? Yeah, because the interferons secrete everywhere, and they bind to receptors, and they induce all these gene products that have a lot of consequences, including fever, chills, nausea, and malaise. That's induced by interferon. It's not the virus doing it. Well, the virus is infecting you, so I suppose in a way it's doing it, but I see it as a host response. It's necessary. It's the consequence of trying to clear an infection. You have to feel badly. Every viral infection results in interferon production, and that's why flu-like symptoms are so common. People think, I have flu-like symptoms, fever, chills, nausea, malaise. A lot of viruses can do that because they can all induce interferon. Next question, how do interferons limit viral replication? Interferons directly inhibit viral translation. Interferons lyse viral particles. Interferon-induced ISGs. Interferons damage cells, or none of the above. I'll give you a moment to think about this. Here's the answer. Interferons induce ISGs. They don't lyse viral particles. They don't directly inhibit viral translation. Interferons bind to receptors and induce genes that have their effects. They don't damage cells directly. The only thing that interferons do is to bind to receptors and then turn on the synthesis of other genes. By themselves, that's all they do. So they have no effect on their own. The innate immune response is also comprised of sentinel cells. And as the word suggests, these are patrolling your body looking for trouble. Dendritic cells, macrophages, natural killer cells. Here's a dendritic cell on the left here. It's called the dendritic cells because it has dendrites that look like the dendrites of neurons, but they're not neurons. They're not neuronal cells at all. They're immune cells. And they patrol our body looking for signs of change. Here's some NK cells in orange, getting very close to a cell. We'll look at how some of these work. Dendritic cells are flowing through you. They're in your circulation. They're in body cavities, and they're also at epithelial surfaces. And there are lots of different kinds of dendritic cells. As you can see here, there are dendritic cells in the blood. There are dendritic cells in tissues. But what you need to know is that they're everywhere because they're looking for something foreign. So here are some dendritic cells below the epithelial lining of the intestine. And they're there sampling materials that are either released from cells or that come into the subepithelial spaces. There are also dendritic cells that patrol other epithelia, skin, eye, vagina, respiratory system as well. And again, all these dendritic cells are sampling the environment. If apoptotic bodies are released from cells, for example, a virus-infected cell, they will take those and they go to the lymph node. So they go from their site where they're picking up material, they go to the lymph node. They go in the lymph node and they present these foreign materials to T and B cells that are in the lymph node. And the T or the B cell will determine if this is foreign or not, using a mechanism that we'll talk about next time. And as these dendritic cells go into the lymph node, they also produce cytokines, which depend on the kind of foreign molecule that they've picked up. And that cytokine also helps the B and T cell to figure out what's going on. We'll talk about that later, but the point I want to mention is that this is, again, dendritic cells are part of the Sentinel system, that they're always patrolling areas looking for foreign material. When they get something, they go to the lymph node, and the B and T cells decide what's going on. And here on the right is a very cool picture of a dendritic cell, which is labeled green. It's very close to a T cell. And here there are blood vessels you can see as well. So this dendritic cell is presumably come in from the periphery, and it's asking the T cell to figure out what's going on with this particular molecule. Here's a summary of that, an overview. So we have virus infecting epithelial cells. The infected cells produce cytokines as a consequence of the virus being sensed by the innate immune system. These cytokines can attract dendritic cells, and if viral proteins and nucleic acids are picked up by the dendritic cell, that's their job, to pick up these things. That plus the cytokine will cause them to mature. They will migrate to the lymph node, and there they'll interact with B and T cells. And if it's a foreign substance that the dendritic cell is brought in from the infected area, then we'll have adaptive immunity. And we'll talk about that next time. Just wanted to make this bridge evident for you today, this bridge between the innate and the adaptive systems. These DCs and other sentinels are key elements of that bridge that take the innate response and bring it to the adaptive response. Let's look a little more closely at dendritic cells. So they are circulating in immature forms. These cells have toll-like receptors on their surfaces. They have cytokine receptors. They have MHC class II in the endosomes, major histocompatibility proteins of the class II, and these are going to be involved in presenting proteins to T and B cells in the lymph node. So again, imagine that this dendritic cell, this immature dendritic cell, is at an epithelial surface that's infected. It can sense virus or virus protein via toll-like receptors, either at the cell surface or by taking them up by endocytosis. Can be activated by inflammatory cytokines produced by the infected cell or by parts of dead and dying cells that are picked up. So these can then produce interferons, which can further amplify the effect of the antiviral effect of interferons. And all of this will cause the dendritic cell to mature and migrate to the lymph node. And a mature dendritic cell looks like this one on the right here. It has now taken whatever proteins it's picked up at the infected site, those little orange blocks, those are taken up and loaded onto MHC class II, a process we'll talk about in more detail next time. These are now displayed on the dendritic cell surface, and here you can see it is presenting one of these peptides to a T cell, and this is happening in the lymph node. So this activated dendritic cell has gone all the way into the lymph node. It presents its prize here to the T cell. At the same time, it's producing cytokines of various sorts, and the kinds produced depend on what kind of antigen has been picked up. And if this is foreign substance, the T cell would become activated and then differentiate and initiate the adaptive response. So that's one of the links between the innate and the adaptive system. The DC is getting things ready in case the virus replication is unchecked by the innate immune system. We start the adaptive response if it's needed. Talk about that more next time. Another component of the sentinels is NK cell. And this is a type of cell that recognizes absence of self. So here we have an NK cell on the left, which is scrutinizing a cell which may or may not be infected. And the NK cell interacts with cells via two kinds of receptors. First, there is a receptor on the NK cell that interacts with MHC molecules. And here the MHC has a peptide in it. And then there is a second receptor on the NK cell interacting with another protein on the cell surface of the target cell. This one is called an activating receptor. So the default is to activate killing of the target cell unless the second receptor is finding MHC on the target cell. So if the NK cell engages the target cell with both ligand receptor interactions, nothing will happen. In fact, many virus infections lead to downregulation of MHC molecules on the cell surface. They downregulate MHC, so the infected cells cannot be detected and eliminated, as we'll see next time. So the NK cell comes along. If it does not sense the presence of MHC on this cell, the activating receptor will lead to NK killing of that cell, assuming that it's infected because there's no MHC on the surface. And yes, there are viral modulators of NK cell activity as well. Otherwise, again, there would be no virus infections. Another important part of the innate system is complement. Complement is a collection of proteins in our serum that is there to recognize patterns and to respond to foreign patterns. And there are several ways of activating complement, and there are several consequences of activating complement. So for example, what we call the classical pathway, antibodies interacting with a microbe, it could be a bacterium or a virus, are recognized by this complement protein called C1. It's actually a complex of a number of different complement proteins. And if these antibodies bound to a microbe are detected by C1, it catalyzes a series of processing events which lead to the production of proteins that can cause inflammation, proteins that can cause opsonization. This microbe could then be taken up by a macrophage and digested inside the macrophage. That's called opsonization. Or can lead to the production of proteins that make a pore in the membrane of an infected cell or a virus, and that lyses the cell. So if the microbe in question here were an envelope virus, it could be lysed by this series of events. Or if the antibodies were binding to the surface of an infected cell, this triggering of complement could lead to lysis. And you can see here there are ways that carbohydrates can trigger the complement pathway. So many pathogens have unique carbohydrates that trigger this. Complement is a series of proteins in our serum which is ready to go once viruses or other microbes are detected. And it's immediately available. There are in fact some kinds of antibodies that are present that would trigger complement without having to go through an adaptive response. So this could be a very rapid response as well. Let's talk about the inflammatory response. What is inflammation? When your cells are infected, as we've said, they produce cytokines and chemokines. These are similar proteins that bind different receptors, but they all have immune activating effects. All of this leads to what we call inflammation. Inflammation is a response not only to virus infection, but to also other diseases like cancer, heart problems, Alzheimer's, pulmonary arthritis. Even if you exercise very hard, you will get inflammation in your joints. I guess if you study too hard, you can get brain inflammation as well. This guy's brain is red. But it's not just caused by virus infection. And there are 4 signs of inflammation: redness, pain, heat, and swelling. And the Roman Celsus recognized this in the 1st century AD. He called it rubor, dolor, calor, and tumor, the 4 classic signs of inflammation. And these are produced by increased blood flow, increased capillary permeability, the influx of phagocytic cells, and tissue damage. And these effects are all caused by cytokines and chemokines that are induced by not only infections, but by these diseases, and as I said, exuberant exercise as well. That's the inflammatory response. It is not necessarily caused by infection, but it can be. Now, the cytokines that do this are divided into 3 classes. We call some pro-inflammatory, IL-1, TNF, IL-6, IL-12. These are classic pro-inflammatory cytokines that induce the symptoms of inflammation that we just talked about. We have anti-inflammatory cytokines. Their job is to come in and suppress the pro-inflammatory cytokines, because imagine you make pro-inflammatory cytokines in response to a virus infection. You keep making them even after the virus is gone. They may last. You don't want that. You want to be able to suppress them. So it's a control system. And then chemokines, their job is to recruit immune cells into the infected area. So they cause an influx of, say, phagocytic cells, which is in part leading to the swelling typical of inflammation. So these cytokines would be made locally initially, right, if you have an infected epithelial barrier, but they will enter your circulation. You can't prevent that. And they will have global effects. They will cause sleepiness, lethargy, muscle pain, no appetite, nausea, flu-like symptoms. That's why a localized viral infection can have global effects, and why if you have flu, you can have nausea, because you're making tons of cytokines, they're going all over and engaging receptors on various tissues and producing these effects. And here is a picture that's illustrating this. Here we have some pro-inflammatory cytokines being produced, maybe as a consequence of influenza infection. They get into the circulation, they can cause fever, fatigue, and sleep. They get in your brain. And importantly, some of these CSFs produce colony-stimulating factors, go into the bone marrow, and have it make more lymphocytes. The precursors are all there. They can divide and make more lymphocytes to get into the circulation to deal with a infection. In this table, we mentioned chemokines as being able to recruit immune cells. Let's look at how that can happen. So on the left, we have a diagram of how cytokines work. There are receptors for cytokines on host cells, and they initiate signaling cascades, just as interferon binding to cells initiate signaling cascades. Transcription factors go into the nucleus. They stimulate the production of other gene products that mediate the effect of cytokines. On the right is how a chemokine might work. So this is the endothelial barrier. It's part of a blood vessel. And let's say this particular lymphocyte is in the blood, and there's an infection near this capillary. This lymphocyte needs to get into the infected area to have its effect. And what happens is there are signals on the endothelial cells that tell the lymphocytes to start adhering to the endothelium and eventually migrating through it. As I mentioned previously, immune cells can do that. They can squeeze between these junctions. This rolling activation and adhesion is mediated by receptor interactions between the cell, in this case it's a neutrophil, and the endothelium. You can see here there are adherents mediated by adhesion molecules, selectins and cell adhesion molecules here. And here we have a chemokine being produced, IL-8. So again, there's some infection below that has induced the synthesis of a chemokine. This is binding to a receptor on the neutrophil. That leads to the production of an integrin that then will interact with a molecule on the endothelial surface. So the chemokine is inducing events to allow the neutrophil to attach and eventually migrate through to the infected area. So that's what these proteins are doing. Of course, viruses encode countermeasures of cytokines, and there are different sorts as shown here. They encode proteins that interrupt the production of cytokines. They interfere with their synthesis or any steps needed to make functional cytokines, modifications and so forth. They can interfere with their action. Some viral genomes encode homologs of cytokines. They make proteins that look like cytokines, but they're not. They bind to cytokine receptors on the host cell, but there's no signaling resulting because it's not the right protein. And therefore, the consequences of cytokine binding does not occur. They can also encode soluble cytokine receptors. So these will bind soluble cytokines and neutralize them. It will prevent them from binding their membrane-bound receptors. And some viruses also encode proteins that alter the signaling pathway. Remember, the cytokine binds a receptor and it initiates a signaling pathway. Virus proteins can interfere with that signaling to block all the effects of cytokines, which are antiviral. We haven't really talked about the antiviral effects of cytokines, but as a whole, different kinds of cytokines are produced in response to various virus infections. They have antiviral effects, including increased blood supply to get rid of viruses, move them elsewhere, clear them, increased phagocytic cells moving into the area to help clear an infection and so forth, and viruses can interfere with that. Inflammation is actually good. It sounds bad, the swelling and the heat and the pain and the redness, but it's actually good because it signals that the innate response is engaged. Cytopathic viruses, viruses that kill cells, they cause inflammation because they kill cells. And killing cells, making cells break up so that the pieces can be picked up by dendritic cells and other sentinels is good because it gets the innate system moving. Those DCs make cytokines, and those cytokines start the inflammatory process. And that's why viruses that kill cells encode proteins to modulate the immune response leading to inflammation. They can modulate cytokines, for example. And these include adenoviruses, herpes viruses, and pox viruses. They infect, they cause cell damage, and you get a good immune response because the DCs are picking up pieces of dead and dying cells, and they're responding by producing cytokines and chemokines that cause inflammation. However, not all viruses stimulate inflammation, and that's not good for us. These are typically what we call non-cytopathic viruses. The cells are infected, but they're not damaged. There's no apoptosis. There's no cell death. And as a consequence, it's hard to sense these. It's hard for DCs to sense that these cells are infected because they're not breaking up in a way that the DC can sense them. So we have low innate responses, and because of that, the adaptive response is poor as well. A good innate response is essential for a robust adaptive response. Because of this, these kinds of infections, non-cytopathic viruses, are not efficiently cleared. They often cause what we call persistent infections, infections that last either a long time or the life of the host. Now we're going to talk about those later on in a separate lecture. So the lesson of all this, the classic inflammatory response, calar, tumor, dolor, rubor, that reflects how the innate and adaptive responses are communicating, because those effects are produced by soluble molecules, cytokines and chemokines, that are stimulated by sensing of an infection by innate immune responses. If we don't have an inflammatory response, we're going to have an ineffective adaptive response, and it's going to be difficult to clear the infection. And this is one of the reasons why we put adjuvants in vaccines that do not replicate. We'll talk about this more later. But there are some kinds of vaccines that are inactivated, or subunit vaccines. They don't replicate, so they don't kill cells. So we don't get a good innate response, and you don't get a good adaptive response because there's no inflammation. We add chemicals called adjuvants to those vaccines, and what those chemicals do is they stimulate innate immune responses, sometimes by binding toll-like receptors. So they mimic activating the innate response, and you get a better adaptive response as a consequence. As you might guess from what I told you before, not all inflammation is caused by infection. You can exercise and have inflammation in your joints. There are lots of ways to stimulate the productions of cytokines and chemokines. I want to show you this great example of how the way we deliver smallpox vaccine causes inflammation, which increases the immune response to the vaccine. And it's a perfect way to summarize what I've told you. So the smallpox vaccine, which again we don't get anymore unless you're in the military because the virus has been eradicated, it's delivered in this bifurcated needle, which is made specifically for this purpose. It holds a drop of smallpox vaccine. Then what they do is they scrape the surface of your skin with this, the epidermis. So it causes injury. And that injury alone causes inflammation. It's another example of how you wouldn't have to have virus here to cause inflammation. The virus, of course, is going to infect the epidermis and cause inflammation on its own. But in fact, the scraping of the skin with this needle makes this vaccine work really well. It's been shown in animal models. If you don't scrape, you don't get as good an immune response. And the reason is injury is detected and results in the productions of cytokines and chemokines. They cause inflammation. Here we have some immune cells coming in. The area is getting red. It's swelling. All of this is going to pick up viral proteins made by dead and dying cells. They're going to bring them back to the lymph node. And because it's a good inflammatory response, the adaptive response is going to be great as well. Really important component of the efficacy of this particular vaccine. So to summarize what we've talked about today, we have a virus infection which is then sensed by the innate immune receptors, the toll-like receptors and others. The infected cells produce cytokines and chemokines, including interferons. Those attract dendritic cells, which sample the products of dead and dying cells. The dendritic cells go to the lymph node, and there they will interact with the adaptive immune system, essentially displaying what they've picked up at the infected cell site and asking, is this foreign? And if so, what should we do? And at that point, adaptive immunity takes over. And that is going to be the topic of next lecture, where we consider what happens after the innate immune response. And don't forget, all viruses must encode at least one regulator of everything we've talked about today, intrinsic and innate defenses. They interfere with sensing of viral products, the production of interferon, transduction occurring after binding of interferon to receptors, cytokines and chemokines, their production and their signaling. They can interfere with NK cell killing, they can interfere with dendritic cell function, and they can interfere with complement. Every step of the way, viruses interfere with intrinsic and innate defenses, and you'll see next time when we talk about adaptive defenses, they interfere with that as well.